Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the basics of antiviral immunity, essentially what our cells do in the face of a viral infection. Now, throughout evolutionary history, our bodies have developed a wide range of defensive measures to try and protect us from a wide range of communicable infectious diseases, including viral diseases. And so some of the defensive mechanisms are general, applying to a range of infectious agents, whilst others are slightly more specific. And so some general defense mechanisms include physical barriers, such as our skin and mucous membranes, which protect underlying organs from infection and act to physically stop infection. In addition to this, our mucous membranes have an additional mechanism of defense where they can trap foreign materials in mucus, which is then cleared away by other body processes. Other mechanisms to reduce infection include secretions such as stomach acid. Anything that is ingested will be subject to the harsh environment of the stomach, which is capable of neutralising various threats. However, we aren't always able to completely reduce the threat of infectious agents, and nor do we have to. Remember that for an infection to take place, there needs to be an infective dose, and so the body only needs to neutralise threats so that they fall below this threshold. Now, in addition to these broad defense mechanisms, we have some that are more specialised and focused, and these include things like antibodies, T cells, natural killer cells, macrophages, and interferons. Now, in order for these systems to function and defend against viral infection, they first need to be able to detect the presence of the viral pathogen. And so we need to ask the question, what is it that triggers an antiviral immune response and kickstarts the immune system to defend against a virus? Well, it all revolves around what we call PAMPs, which stands for Pathogen-Associated Molecular Patterns. These are protein structures on pathogens, including our viruses, which are antigenic, meaning they are able to be recognized by our immune system. And for our immune system to recognize these PAMPs, cells in our immune system use PRRs, or Pattern Recognition Receptors, which, as the name suggests, recognizes PAMPs on the infecting pathogen. Now, one of the interesting things about these PAMPs is that they aren't just expressed on the surface of the pathogen. They can also be expressed on the surface of a host cell that has been infected and the virus is actively replicating in. Essentially, as the hijacked cell starts to produce viral proteins, some may be integrated into the plasma membrane of the host cell, acting as a PAMP. So let's have a little bit more of an in-depth look at these antiviral mechanisms. And we'll start with our humoral response, which tries to stop virions whilst they are in the extracellular stage of infection, so when they are outside a cell and our body is able to generate antibodies, which has a number of effects on these virions. Firstly, the antibodies will attach to PAMPs on the virions, and this will block the ability of the virion to attach to the target host cell receptor, stopping them from ever entering the target host cell. And I guess a good example of this is mucosal IgA, which is found in both the respiratory tract and gut, acting to protect two key sites of entry into our body from potential pathogens. Now, in addition to blocking host cell attachment, antibodies can also aggregate virions together using the long stem of the antibody. And what this does is it allows the antibodies to clump all the virions together, making it easier for our macrophages to remove large numbers of virions at once via the process of phagocytosis. Now, that rounds out our humoral response, but we also have the cell-mediated stage of antiviral immunity. And what this does is it allows us to react to the intracellular stage of infection. And we need this because at some point there might be a failing of our humoral response, and we need to act on virions that have, for whatever reason, bypassed our initial defense mechanisms and hijacked our own cells. Now at this point, if our host cell is able to determine that it has been hijacked, it will release various cytokines, including interferons, into the environment. Now these signalling proteins are used to alert cells in close proximity that they are under attack. In addition to this, it will signal for neighbouring cells to switch on various interferon simulated genes, which will put it in an antiviral state. Now, as you can imagine, this has a number of knock-on effects. Firstly, it limits protein synthesis within the cell, whilst also inducing fever across the host, and this helps to destabilize the virion. And this is why we have a high temperature during viral infections. It's one of the defensive mechanisms our bodies use to negatively affect a virus. Another knock-on effect is that the cell can display altered surface proteins, which can either be host or virally derived. Ultimately, these surface proteins signal to the outside world that something is wrong with this particular cell. This then allows other aspects of our immune system to come along and mark the cell for destruction. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.